So looking at a table and then watching one more animation. So we're looking at the comparison between the nervous system and the endocrine system. Nervous system is there for fast on, fast off. If we try to run our bodies 24 hours a day, run everything with the nervous system, you'd be eating all day long. You would not be able to do anything but consume food to make ATP to carry out the activities of the nervous system. So you need something that can control the body's main organ systems, but have it available in the circulatory system to keep things running while the nervous system has already turned off. So endocrine system is what the body came up with. Get things that can be distributed by the blood, can last longer. Nervous system, if you remember, neurotransmitters, they have a checklist of what they are, how they get to be established as a classic neurotransmitter. One of those things is there's an enzyme to break them down. So the nervous system, you want it turned on, and then you're going to have something right there or close by. Or if you want to have widespread effects, then you let it go into the circulatory system. But if you remember acetylcholine, that is going to control one organ or one part of one organ. It's got an enzyme that's right there at the postsynaptic membrane to break it down. It's got the same enzyme in the synaptic cleft. It's got it in the interstitial fluid. It's got it in the blood. Acetylcholine is going nowhere. So fast on, fast off. So this is a wired system, but the wires are different than the wires that we think about in electrical circuits because they're gaps. So at the end of a wire, you have a chemical that's released that has to diffuse to the next wire. This is a wireless syndrome, but the receptors that are existing for a lot of the endocrine system can be the same receptors for the nervous system. The hormone that comes out in the endocrine system can stimulate the same postsynaptic neuron that a presynaptic neuron is releasing neurotransmitter to stimulate. So via the blood, you continually stimulate that postsynaptic cell without energy being used up by the presynaptic neurons. So neurotransmitters go into very specific locations called the synaptic cleft. We're going to learn about some exceptions. You've already studied the autonomic nervous system. You know that autonomic neurotransmitters do diffuse away from the synapse. And that's why they have widespread effects, especially that sympathetic nervous system. So they can stimulate cells out of the localized area. But hormones are designed exactly for that purpose, to stimulate widespread. So anywhere there's a receptor, then the hormone can have an effect. The synaptic cleft is very small, so the effects of the neurotransmitter are going to be localized either in that synaptic cleft or maybe diffused to a neighbor. Occasionally on the autonomic sympathetic side, it's more widespread effects. But usually over a small distance in that synaptic cleft. And of course, large distances by the blood. The specificity, the neurotransmitters like acetylcholine, very specific as to what they're going to stimulate. And they're going to be released and then degraded in a very discrete location. The endocrine system depends on where the receptor is located. So the same hormone may stimulate various parts of the body. You think about your fight or flight experiences. You suddenly see more, your peripheral vision increases, you may start sweating, your blood pressure goes up, your heart rate goes up. You have all these things that are happening all at once, and that's due to the recruitment first by the nervous system, but then it's maintained as you're running away from the saber-toothed tiger by the endocrine system. The turn on, turn off, millisecond, so 1/100th of a second, and that action potential is gone already. But with the endocrine system, it may last for minutes, it may last for hours or days. Can be very long. The nervous system is precise, with the exception of the sympathetic branch of the autonomic nervous system. It's rapid, and so you turn it on fast, you turn it off fast, and you get very precise communication with postsynaptic cells. Here it's less precise, but if you think about the cells that are responding having to have a receptor, it's not an indiscriminate recruitment of those cells. So the neurotransmitter can only bind to specific things, and it may fit in areas, but if it doesn't have the charge or the exact shape, then it's going to diffuse off and you're not going to get the postsynaptic cell stimulated. Oh, it worked. Somebody, hold on, somebody turn the sound down again. I don't know why they don't use that. There's a volume control right there. Okay. Oops, I have to be in slide mode.
Bind to receptor proteins and start a process that affects cell functions. Hormones can be grouped into two general types, hydrophilic hormones and hydrophobic hormones. Hydrophilic hormones do not cross the lipid-based cell membrane. Instead, they bind to surface receptors. The interaction of the signal and its receptor will, in turn, generate the target cell's response. Binding of the signal causes a conformational change in the receptor. As the receptor changes shape, it will interact differently with nearby molecules. For example, the receptor tyrosine kinase will respond to hormone binding by phosphorylating the next molecule in the response sequence. The activation of molecules within the cell by the binding of the hydrophilic hormone on the surface can generate a rapid and large-scale response within the target cell. This is a little like the mousetrap game, where a series of events cause a reaction some distance from the original stimulus. The target cell receptor for the hydrophobic hormone is found within the cell, either in the cytoplasm or in the nucleus. Because of their chemical structure, hydrophobic hormones may need a carrier molecule to travel through the bloodstream, but are able to pass through the lipid layer of the cell membrane without difficulty. Just as a hydrophilic hormone does, the binding of the hydrophobic hormone to its receptor causes conformational changes that change the receptor's activity within the cell. In this example, the hormone receptor complex is formed in the cytoplasm and then moves into the nucleus. Hydrophobic hormones affect protein synthesis within the cell. They may either increase or inhibit the transcription rate of specific genes. Because of their direct effects on gene expression, hydrophobic hormones generally have slower, more long-lasting effects on their target cells than hydrophilic hormones have. Aldosterone provides a good example of how a hydrophobic hormone can cause a profound change in cell function. Aldosterone is released in response to a drop in blood pressure, causing the kidney to retain more water and sodium, which causes an increase in blood volume. This is an important mechanism in the homeostasis of blood pressure within the body. To do this, aldosterone stimulates increased transcription of a specific gene in the nuclei of kidney tubule cells. This gene codes for a protein that becomes a sodium-specific entry channel in the cell membrane. By increasing the amount of the sodium channel protein's messenger RNA produced, aldosterone increases the number of sodium channels in the cell membrane. The increase in sodium retention creates a concentration gradient that allows the retention of more water as well, thereby increasing blood volume and blood pressure.
Okay. I couldn't remember if I showed this to you already. I think I did, but I wanted to just make sure to point out to you that your hydrophilic versus hydrophobic, you get a quick response with the hydrophilic because you have the G protein complex inside that takes that first message and creates several other messages potentially inside the cell. So you can activate various enzymes in the plasma membrane and then get various second messengers, and each of those can stimulate a protein kinase, and those can then phosphorylate multiple proteins. And we did run through which has a longer half-life, right? Which type of hormone, the steroid, hydrophobic, or the hydrophilic hormone has a longer half-life? Hydrophobic because it has a carrier protein. Okay, and I did tell you, did I not, that one of the statements that aldosterone is increasing water reabsorption is not really correct. It's influencing sodium reabsorption and water. Then that sodium changes the water concentration in the blood, and water will move down its concentration. So it's the water movement is secondary to the sodium reabsorption. Okay, so we're going to go to integumentary system. Why isn't it coming up? Okay, that's the way to do it. Okay, so integument or skin is the largest organ in your body. People don't think of it as an organ, they just think of it as a covering and kind of inert. But it's got receptors in it that tell you about your environment, and without those receptors, you end up doing a lot of stupid things. If you've seen programs about people who are born without sensory receptors, they'll jump off the jungle gyms. They'll go into hot showers and get burned, and they have, or they'll cut themselves. Babies, when they're teething, they'll bite through their lip. They'll bite off pieces of their tongue. And they have no idea that they're doing that because they have no pain receptors. They have no thermal receptors. So without those, you don't know when you're put in potential danger. So integumentary system is broken down into the cutaneous membrane and then the accessory organs that go with it. They're essentially all coming from cells that are from your epidermis, and then they'll be modified to create things like fingernails and hairs and sweat glands. The cutaneous membrane, epidermis, that's your stratified squamous, keratinized tissue, so epithelial tissue. The keratinization makes it tough, and you can scratch it and you don't get cells under your fingernails. You also have stratified squamous in your mouth, inside of your eye, in your urethra, your vagina if you're a girl, and the outer part of your anus. When you touch those with your finger, hurts. go inside your mouth and scrape it, the cells come off underneath your finger. You can take a DNA swab of the mouth and you'll get nuclear material from the cells on the surface of the mouth. But you cannot get that from your skin. So when you watch TV programs and they say, oh, epithelia there, and they're going to get DNA out of that, and no way, Jose. Unless the whole epidermis came off, or you cut down into the live cells, no way you're getting DNA from what is exfoliated. That's dead. There's no nuclear material. There's no organelles. No nothing there. Okay, what is the function of the epidermis? It's a barrier. The whole skin is a barrier, but the epidermis is designed to be the best part of that barrier. Because it's keratinized, it's tough. You don't get cells just flaking off with friction. You don't get water coming through, and you don't get water going out unless the water is going out through an exocrine gland. So you're controlling the amount of water that you're losing. One of the dangers of burns is you lose the epidermis and people dehydrate very quickly. So people with extensive burns, they're always hooked up to an IV to make sure they don't dehydrate to death. So permeability is one of the functions of that barrier. Chemical trauma, friction forces, other kinds of trauma, chemicals. You get shampoo in your eyes where it's non-keratinized and there's no dead cells on the top. Hurts a lot. You get it up your nose, it hurts a lot. You get it in your mouth and it's not so bad there, but it doesn't feel that great. Get it on your skin, your scalp, no problem. Vitamin D3 is, its synthesis begins in the skin. It isn't complete till it goes to other places. We'll talk about that when we talk about hormones. The sensory receptors in the epidermis are just for light touch. You don't have the pain receptors are underneath the epidermis. Temperature and pressure receptors are actually in the dermis. So just light touch, light pressure that's actually in the epidermis. You have special cells in the immune system that hang out in the middle layers of the epidermis. They're there to ensure that if bacteria get through the dead cell layers, that they're not getting any further. They're not getting through the basal lamina and into the underlying tissue where they could get into the blood vessels. Epithelia, all epithelia are avascular, and that includes your epidermis. No blood vessels go into the epidermis. They're all below the basal lamina, and the cells are getting their nutrients via diffusion across the basal lamina. 2 layers of the dermis. The upper layer we'll see later is loose connective tissue, so that cells can wander through blood vessels, nerves, lymphatics, can be right close to the basal lamina so that things can diffuse. The reticular layer, you get a dense, irregular connective tissue. You're back to a better barrier again. You'll have some fat in that area, but most of the lipid reserves are in what's called the hypodermis, and that is not part of the integumentary system. So I'll be hammering that into you. Hypo is below the dermis. We're talking about integumentary system, which is dermis and epidermis. That reticular layer is where you have the deeper touch. You have more. You have the vibration senses there. You have the thermoreceptors there. You also will have temperature, vibration, pressure, deep touch and vibration. Pain will be right below the epidermis, and there'll be pain receptors that are down deeper. When you pull somebody's hair, it hurts. It's due to pain receptors that are associated with the hair shaft. Accessory, we have our hair follicles, and then we have our fingernails and toenails, and then we have exocrine glands. And these are all based on, they form from the epidermis invaginating and the cells growing down. And then they'll turn into ducts for sweat glands and secretory cells for the sweat glands. They'll also change into cells that will produce the hair that's going to grow out of the hair follicle. And then you'll have cells that will modify and form very highly keratinized, even more keratinized than your skin. Your hair is more keratinized than your skin, and your fingernails and toenails are even more keratinized than your hair. Then you take your fingernail and you press on the pink part of your fingernail. You can press really hard and it doesn't hurt. You feel the pressure, but you're not screaming ow. You take that same pressure and you put on your skin, you might say ow. It doesn't feel good at all. You come with just light pressure and go to the white part of the fingernail and just lightly press on that. It hurts a lot. That is only partially keratinized. So this is keratinized, this is keratinized, this is the most keratinized, and this is only partially keratinized, that white part. So that is less protected, and that's why it's very sensitive at the root of the nail. Okay, the parts of the skin, we have epidermis on the top. And if you look at the cells at the top of the epidermis, there are no nuclei there. They are also different colored than below. We don't have purple cells under beige cells in our skin. Our skin is kind of beigey. But underlying the top layers, you have live cells, and those you can pick up more stain with those because they have the organelles there. The top layers are dead. They're also compacted because most of the water has come out of the cell. And in certain areas of your body, you have a thick enough epidermis that you can see the transition from the live cells to dead and dying cells to dead cells. And they're going to name the different layers. We'll see on a later slide. The sensory receptors can be lamellated, meaning you have lamella, you have layer upon layer of epithelial cells and a nerve in the center. So you've got to compress those epithelial cells to stimulate the nerve. So these are going to be vibratory receptors and pressure receptors. We have a tactile corpuscle, usually that is right up in this little dermal papilla here. And then we'll have free nerve endings for pain. We'll have free nerve endings associated with the hair shaft. Thermal receptors are there, other vibratory sensors are there. They're going to be, we'll cover those in depth when we get to the nervous system. We've got blood vessels, we've got arteries going to these structures, but all the arteries stop at the reticular layer. They do not go into the epidermis. Epidermis is avascular. Veins going back. The large arteries and veins are in the subcutaneous, hypodermis. Another name for that is superficial fascia. We talked about that in the earlier chapter. So this is actually not part of the integumentary system. It's there to support the skin. It's supportive via nutrition. It's supportive via this fat padding. So it's there. It does have a purpose, but technically it is not part of the integumentary system. It's always studied with it because of its supportive nature, but it is not part of the integumentary system. So the cells of the epidermis, they are going to be, most of them are going to be keratinocytes. Keratin is the intermediate filament that I specifically told you about when we covered the cell. Those intermediate filaments are tough. There are a lot of them. They're called tonofilaments as they're being formed. And those will go to the lateral sides of the cells and they'll form desmosomal attachments with neighboring cells. So when your epidermis flakes off, it comes off in chunks because those desmosomes keep multiple cells together. So when you get dandruff, you see flakes on the person's shoulder or flakes in their hair if it's stuck up there. You wouldn't see dandruff if it was individual cells that were coming off. But because the cells are stuck together by desmosomal attachments, you actually see them. The keratinocytes are coming from stem cells at the basal lamina, just superficial to the basal lamina. The stem cells divide, they replace the stem cell, and then they create a keratinocyte that will move one layer up. There are 5 layers of skin that you can identify in thick skin. There are 5 layers in thin skin as well, but you can't identify one of those 5 layers. And that's the layer where the cells are transitioning to be dead. So they're dying, the nucleus disappears, the organelles disappear. When you have thick skin, you have multiple layers of these dying cells. So when you fix the tissue and put it on a slide and fix it, you can actually see those cells that have lost their organelles. In thin skin, you have to have a transition zone as well, but you don't have multiple layers, therefore you don't see it. So 4 layers can be identified. So you can find 4 layers in the thin skin, but you can find 4 layers in your thick skin. Where is your thick skin? Palms of your hands and soles of your feet. Those areas where you get the most friction. Melanocytes, those are the ones that produce the color to your skin. The ones that react to UV radiation and give you a tan. These are pigment producing cells. It's not the melanocytes that are creating the color of your skin. The keratinocytes actually chomp on the melanocytes and phagocytose part of the cell, bring it into themselves, and then condense it into a little pigment granule. We'll talk more about that later. So the color is actually transferred, the pigment is transferred to the keratinocytes, which are much more numerous than the melanocytes. So body of the melanocyte is down there where the stem cells are. But the melanocyte has long processes that extend upwards into more superficial layers of keratinocytes. So it can serve multiple layers of keratinocytes, serve up the pigment to them. The Merkel cells are the only sensory, they're not, so you'll probably see in some books they won't call it a cell, they'll call it a disc, so that you know that it's not a complete cell with a nucleus and organelles and all of that. It's acting as a sensory receptor and it's hooked up to myelinated nerve. This is the only sensory receptor that is in the epidermis. All the rest are in the dermis. You also have this immune cell there. It more correctly nowadays will be called a dendritic cell for its shape. It has long cell processes that look like the dendrites of neurons, but it's not a neuron, it's an immune cell, it's more like a macrophage. It's an antigen-presenting cell. These are fixed macrophages that are there to look for anything that gets through the dead cell layer. It will then process and present antigen to other white blood cells so that those white blood cells can move off and look for anything that might have gotten through the barrier where the dendritic cell was not sitting and prepared to handle the pathogen. Langerhans is a guy's name. You're going to find Langerhans cells in the pancreas, islets of Langerhans in the pancreas. You're going to find other things identified with that eponym, and that's what gets confusing, and that's why they're trying to get away from those people's names. But Langerhans, it's in your textbook. It's probably going to be in the textbook for 20, 30 more years. It might be there longer than that. Always you come across it, even though they're, for 20 years they've been trying to get you to change to say dendritic cell only, but still put Langerhans cell in the textbook. These are the 5 layers that you can identify in thick skin. So at the bottom, the deepest layer right next to the basal lamina, you have the stratum germinativum. It is where the cells undergo mitosis. They can undergo mitosis a cell layer or 2 up as well. So some of the keratinocytes that are produced by stem cells at the stratum germinativum also undergo mitosis up above that. But the stem cells will be located in that first layer. It also can be called the stratum basale. So that is telling you that is the basal layer of the epidermis next to the basal lamina. Either way of describing is perfectly okay. So stem cells divide and you get the keratinocytes formed that will move up and start to form these tonal filaments, the tonal fibrils that form the cytoskeleton of the cell. They also will attach laterally to form desmosomal attachments with the neighboring cells. In the stratum spinosum, you'll have Langerhans cells, you'll have melanocyte cell processes, and maybe part of the cell body, but most of the cell body is down here with the stem cells. We'll see that on a later slide. With the production of the tonal filaments, also the cell begins to form granules called keratohyalin granules. You see that up here, keratohyalin being produced. Actually, it's produced in a stratum spinosum, begun to be produced in a stratum spinosum, but it doesn't take up, the granules don't take up much space. You can't see them very easily because there are not a lot of them. And when you take the tissue and you fix it by running it through a series of alcohol dilutions to make the stain stick, then you get the water coming out of cells. In the stratum spinosum, the water leaving the cell causes them to shrink like the little cremated red blood cells, and all the internal cytoskeletons poke outwards. They also poke outwards because they're attached to their neighbors. So you get a very spiny looking cell because most of the water has gone, but the little desmosomal attachments keep some of that surface of the cell pulled outwards. When you get enough of the keratohyalin granules inside of a cell, even though you're dehydrating and the water's coming out, the granules keep the cell shaped during that running through the series of alcohols. And so up above, you have a granular layer called stratum granulosum. Now you have enough granules to maintain the shape of the cell, even though you're dehydrating the cell, and also enough of those granules that you can actually see them. So those cells will look granular down below. In fixed tissue, they actually look spiky, and so that's called stratum spinosum. In the stratum granulosum, you're also secreting I guess it's on a later slide. In this layer, you're secreting a waterproofing around the cell so that you don't lose water from the underlying tissue and you don't gain water from the external surface. But because water can't get into the cells, then nutrients aren't getting into the cells either. And so then the cell starts to die. The nuclei, the other organelles disintegrate, and you're left with a rather puffy looking cell that hasn't been compressed by other cells pushing from below, other dead and dying cells. So in the thick skin where there are enough of those dying cells, when you fix the tissue, put it on a slide and shine light from below, light comes through those cells. Where the nuclei and organelles are there below, light doesn't come through. When the cells are compacted enough above that transition zone, because water has left and other cells are pushing on there from below, they just flatten out, then light doesn't come through either. So at that transition zone where the cells are dying, you get light coming through. That is called stratum lucidum. Lucidum means light is coming through. The light goes on. If you're good with English, you know that when somebody says, oh, you have very lucid thoughts, that's when the little light bulb in your head is going on and you're thinking clearly and expressing your thoughts clearly. Stratum corneum. Corneum means it's dry, it's scaly, horny looking. That's not in the vernacular term, but that the little pieces of skin come up and they create things that look like horn. When you have thick skin, you may have up to 50 plus layers of these dead cells. The thick skin is designed to resist frictional forces, so you have a lot of those layers. So stratum corneum is your dead cells. They're all waterproof. Very little water will get through there. The stratum lucidum looks kind of glassy. That light comes through. You don't see speckles anymore. You don't see any organelles anymore. The only thing that can penetrate the skin, what do you think can penetrate the skin? Yeah, needles will.
Water.
Water doesn't go through. Water comes out if you have a sweat gland. But what kind of lotions? Yes, vet students. What kind of you're saying topical solutions, and then you said lipophilic. So DMSO, what is it? Dihydromethyl sulfide? No. Dimethyl sulfoxide. Yeah. Dimethyl sulfoxide is extremely lipophilic. In veterinary medicine, the horses don't like needles. So if the horse injures themselves, they'll put the the steroid in DMSO and just rub it on the outside of the skin. The DMSO penetrates and pulls the medication in with it. And anything that is combined with DMSO, anything on the surface of your body that hasn't been cleaned off, will also penetrate with DMSO. And veterinary DMSO is not the same as human grade DMSO. You can't buy human grade DMSO. So people who had arthritis used to run to the vet and say, "Can I have some DMSO?" And they're thinking, "Oh, well, you're going to go treat your horse." So they give them some, and then they go home and they put it on their skin, and then any dirt, bacteria, other things that were on their skin would get in and would get really sick, and the doctors are all confused. What's wrong with this person? All of a sudden they're really bad. What are you doing? I don't know. I don't do anything different. And then finally they find out, well, they had access to DMSO. And that was the problem. So stop using the DMSO. But what else? So corticosteroid creams. You got an itch and you've got hives or you've got some inflammation. You go and get corticosteroid cream, you rub it on. The corticosteroids penetrate and treat the irritation. What else do you wear as a patch? Nicotine patch. So nicotine is lipophilic. What else?
Morphine.
Morphine, I don't know if they use a patch for morphine, do they? I think they usually put it IVs. Birth control patches. You mean like Sabunpa? Oh, that's okay. I don't know about that one. But you have on the TV, just watch estrogen patches to treat postmenopausal estrogen reduction. So progesterone, estrogen, anything that's a steroid hormone will cross. You have these guys treating their erectile and prostate problems with testosterone creams, and then you're not supposed to have females and children come in contact with that. So anything that's lipophilic can get through the skin. Take the epidermis off, you got bumps in the dermis. These are dermal papilla. This is an epidermal ridge that fits into your dermal papilla. Thin skin, the epidermis is thinner, and thick skin, they say the epidermis is thicker. Actually, thin skin and thick skin alone, that's a misnomer. That's misrepresenting the situation. You can have thick epidermal skin and thin epidermal skin. Can also have thin dermal skin and thick dermal skin. So you can combine them in different locations. Where you have thick epidermal, that's where you have the most layers of the stratum corneum. You also can identify the stratum lucidum. So the epidermis is thick where you have the greatest frictional forces. So palms of the hands, soles of the feet. And you'll find that you have fingerprints and you have toe prints. You also have other ridges, but the ones that are best for gripping are the finger and the toe prints. So when you want to run fast and you're barefoot, you grab the ground with your toes. If you don't put your toes down and you try and run on the balls of your feet, usually you slip. So toes are gripping. Fingers are gripping, grabbing something that's wet or slippery. If you grab it with the palms of your hand or another part of your body, it's more likely to slide right through. So on the forearms, no grip at all. Palms, you got a little better grip. Best grip, you can actually lift all that with just 2 fingers. So you've got good grip with your fingers, less grip with your palms, no grip with other parts of your body if it's really slippery. So the ridges are there interlocking with the dermal papilla. Thick dermal skin on your back of your neck, between your shoulder blades, very thick dermis. You have a thick dermis, you lose sensitivity. When you get to the nervous system, we're going to do something called two-point discrimination, where we have calipers, and if you put the calipers 1 to 2 millimeters apart and touch the back of the neck or shoulder, or even the back of your hand, when a person's eyes are closed, they'll say, even though there's 2 points of contact with the caliper, they'll only perceive 1 point of contact. And it's kind of amusing how far apart you have to dial those calipers to get 2 points of discrimination, meaning you say there's 2 points of contact on the back. So we'll do that in class when we get to the nervous system. Thin epidermis, lips. Thin dermal skin is actually most of the body. Your eyelids, you look up at the lights with your eyes closed, you can see the light through the eyelids. So you've got thin epidermis, you've got thin dermis there. Lips, you got really thin epidermis. That's why most lips look a little pink, because you see the blood vessel color through the epidermis. Macaulay Culkin was the first kid or person actually that I ever saw. I thought, why does that little boy have lipstick on? It looks so weird. But then I found out that's not lipstick, it's not makeup, that's just his skin is that cherry color. He still has those big cherry colored lips. Okay. So back to the layers, individual layers. Stratum germinativum or stratum basale is fine. This is where stem cells divide. This is also the location of the cell body for the melanocyte. Stratum spinosum is where tonofilaments are being formed. The most of what the cell is doing is forming tonofilaments, but it's beginning to form the keratohyalin. So acylglucosyl-ceramide, this is also being produced in the stratum granulosum, and that's your waterproofing. That's going to be secreted out over the surface of the cell, and that's what's going to cause the cells to start to die, because nutrients can't get to the cells. When the cell nuclei disappear, the organelles disappear, then if you've got enough layers of them, you see light, and that's stratum lucidum. This involucrin, it's non-keratinized cell wrapper, as well as this acylglucosyl-ceramide. These are both indicating that the cells are on their way out, that they're going to start dying. And then when you get to the dead layer, that's your stratum corneum. Tends to be dry. The only kind of lubrication you have for the skin surface that's coming from your sweat glands, your apocrine and your merocrine sweat glands, and also your sebaceous glands. So sebum coming out is mainly designed for the hair, keeping the hair pliable and not breaking off. But it comes out to the surface of the skin. You get it a lot of it in the hair, and sometimes somebody hasn't washed their hair for a couple of days and they get kind of a funky smell. And then also you don't take a bath regularly, you get a funky smell on your skin, because of the reaction of those secretions with the bacteria on the skin. And you have to have bacteria on your skin, because the bacteria on your skin help to keep it healthy. So some bacteria are bad, but we carry a lot of bacteria on the surface of our bodies that actually keep our epidermis more healthy. Okay, so this is your fingerprint. You actually have sweat glands that are opening in your fingertips. If you rub your fingertips together, you get a kind of a tacky feeling. The more you rub, the stickier your fingers get. And that's when you still have the ability to induce the sweating, the secretion of sweat with frictional activity. When you go and you see grandma, grandpa, probably maybe around 70s or so, in 70s, 80s, 90s, you find that grandmas and grandpas, they're always licking their finger to turn a page because they don't have that anymore. If you touch their hand, the hand doesn't get as wet when you rub it. If the whole hand doesn't get moist when you rub it, then it's usually a sign of something going on with the nerves. But the sweat glands will decrease as you get older. Your epidermis, we'll see at the end of the chapter, changes in its consistency, so does the dermis. Right now you think you're invincible, but you're going to age too. Okay, so skin markings. You probably all have freckles or moles or both. These are hyperpigmented areas in the skin. The freckles are flat. The moles are mounded. Freckles, as far as I know, with UV exposure, they don't get melanoma. You don't get melanomas from freckles, but I might be wrong. But they've always warned you, you go to the dermatologist, he's kind of glancing probably over the freckles, but he focuses on the moles. Every time I went to my dermatologist, he looked at that mole and he said, I cut it off for you. I said, hey, wait a minute, it's not changing. It's been the same since I was a baby, so why are you wanting to bring a knife in? Well, just in case I can remove it for you. Don't you want it off? I said, well, watch it. I can see that one. You look for things I can't see and then let me know if they're changing. But the moles, you want to always have them inspected. If you have moles in the front that you can see, you got moles on the back that you can't see. And you need to have them checked for their change in shape and color, because that's an indication that they are now becoming abnormal. If you look at your wrists, you look at your elbows, you look at the back of somebody's knees, you look at the back of your ankles, you have creases. Then go and find a baby and look at their elbows, their wrists, their knees, and their ankles. There may be one line that's there because of all the chubby little fat and it kind of comes in at a point, but there's not these multiple lines. These are all due to our being able to manipulate objects and move our bodies through flexing. So they're called flexor creases. The hemangiomas, these are birthmarks. Some people have them little bits here and there. Their strawberry ones tend to disappear. They're all related, whether they're strawberry color or port wine color. They're all minor or sometimes major changes in the blood vessel consistency and location. The an area of high numbers of superficial blood vessels give you the strawberry birthmark. And those seem to, as you grow, you kind of outgrow that concentration of blood vessels. The port wine ones tend to stay. They tend to be more on the surface. The blood vessels are already pushing out that surface area. And they don't go away as you get older. Although I have seen some strawberry birthmarks that are there in adulthood as well. Okay, papillary layer of the dermis, loose connective tissue. You want to have space for cells to wander in and out. If your Langerhans cells up here mount an immune response. You want to be able to get the blood cells out of the bloodstream to the epidermis for antigen presentation. You also want to have a less dense area for diffusion to occur. So you have area for small blood vessels, you have areas for your sensory receptors. You also got space for things to diffuse in and out. So there's a lot of cells there, but not a lot of fibers in the papillary layer. You get down into this reticular layer, then you get this very dense, irregular connective tissue. And you got a lot of bundles of collagen, more fibers, and fewer cells there. You have the blood vessels going through that area, but you don't have exchange from arteries and veins to the tissue. It's going to be from the capillaries that are in the tissue. And there's still enough area for capillary and nutrient exchange in the reticular layer. When you get down to hypodermis, it's going to be large blood vessels, connective tissue, and the specific connective tissue there is going to be your fat cells. So, color of the epidermis. Okay, finish this and then we'll take a break. So, color of the epidermis, you look at your neighbor, even if you're both Japanese, Chinese, Asian, it's going to be different color. Now, you compare Haole to Asian, it's going to be obviously different. But if you look at different Haole skins, some are redder and some are yellower. And you look at Asians, also some are redder and some are yellower. Why? The redness comes from superficial capillaries. The yellow comes from keratin. And it's not that they're ODing on carrots and vitamin A, but they just tend to hold more keratin in their skin. The melanin, when you look at somebody who has dark skin, that's not totally due to tanning, although dark skins do tan, but they, even without UV radiation, they stay darker. And then Haole people, you go out in the sun, you turn brown, and 2 weeks later or a week and a half later, it's gone. The difference is not that we have less melanocytes, is that our keratinocytes break down the melanin faster. So we're both making melanin. The transfer is the same, goes to the keratinocytes. The keratinocytes store the melanin, use it for UV protection. But we have, in Haole skin, you got a higher concentration of the enzyme that breaks melanin down. And in darker skins, you have less of that. So with evolution, based on where you originated, if you originated closer to the equator, then you lost some of this enzyme so that you could keep the melanin longer and get more UV protection. If you were closer to the North or South Pole, you're the Germanic and Dutch extraction and English extraction, French extraction, which I am all mixed up, then if you grew up in England, it rains all the time. You think Manoa rains? No, England rains even more. So you're not outside with UV exposure. And so you maintain all this all the enzymes to break down the melanin. So it's based on that, why you have different skin colors. So Haoles have more of the enzyme to break down melanin, and darker skins have less of that enzyme, so they can maintain the melanin in their melanocytes. When you have somebody with dark skin, the white lines show up even more dramatically. You look at a person with really dark skin, the palms of the hands are much whiter than the back sides. You look at mine, you can sort of see a difference between the back of the hand and the palm of the hand. your dorsal surface in anatomical position, things that are dorsal tend to be darker than things that are ventral. And there's a line of demarcation between the extensor surface and the flexor surface. Now, if you say, oh yeah, but